All right, it is October 3rd. This is Biology 105. Thank you all for making it. Today we are going to be wrapping up our discussion on cell membranes, okay, including a brief discussion of osmosis, something that you've already spoken about in your lab section, but I'm going to reiterate it here and maybe explain it in a different way that might make more sense to you. All right? Otherwise, yeah, that's where we are. Any questions before we get started? Okay, let's just get right into osmosis. I'll move through it pretty quickly, but slow me down if you have any questions. Okay. Water movement across a membrane that only allows water to move. That's what we're talking about with osmosis. Now, osmosis is an example of diffusion, which is an example of passive transport, right? It's all about how molecules move from areas of high to areas of low concentration. But when we're talking about osmosis, we focus more on the concentration of whatever is dissolved in the water. We focus more on that than on the concentration of water molecules. So really quickly, look at this U-tube right here. There we go. It's a glass tube with a semi-permeable membrane at the bottom. Semi-permeable membrane at the bottom, all right? There are a bunch of sugar molecules on the right side. There are fewer sugar molecules on the left side. The side on the right must therefore have a lower concentration of water because there are sugar molecules in there, and those molecules take up space. So water moves from an area of high water concentration to an area of lower water concentration. And it will cause the water level to actually go up because doing this increases the concentration on the left side and lowers the concentration on the right side because concentrations are expressed in, say, grams per milliliter, something like, or milligrams per milliliter. So if you increase the number of milliliters, increase the denominator, you decrease the concentration. All right? We cool? Sometimes people are, confused about concentrations and what they actually are. So I thought I would just check with you real quick. All right, solid. Why does any of this happen? Okay? There's a lot of complicated aspects to osmosis. Okay? It's not as simple as I'm going to explain it here, but I'm going to go into more depth than you would usually expect from a class that discusses osmosis. Some particles are osmotically active. Some particles are osmotically inactive. If this was entirely a discussion based on concentrations of water molecules, then it doesn't make sense that any particles would be osmotically active or inactive, because everything would be dependent on concentrations. So what we're really talking about here is the amount of kinetic energy that the water molecules have on either side of the tube here, okay? So let's zoom in to the selectively permeable membrane right here. The side here has higher solute concentration. This side over here is lower solute concentration. All right, solid? These particles, yes?
I got a question. In the example you used above, does gravity have, can this osmosis force fight, gravity itself, how the water is on different levels?
It can, but in this U-tube, the reason that these glass U-tubes are used to demonstrate osmosis is both sides are under the exact same pressure from gravity. So osmosis can be powerful enough to draw things against the force of gravity. Right. Yeah, awesome. Good, but when we're trying to do osmosis discussions around solute concentrations, we want to hold everything else constant. So this allows us to hold the effective gravity constant. Yeah, awesome. So check this out, right? These water molecules on the right side of the selectively permeable membrane, they are interacting with the solute particles, and that's the key, okay? These water molecules on this side of the tube, overall, if you took the average speed of each water molecule, these would be moving around less because they're spending some time interacting with solute particles. So if this chair is a sodium ion, right? And I'm a water molecule. And I've got my negative region here and my positive region over here, all right? I'm just zipping around. I'm moving in completely random directions. It's Bayesian motion. That's how that's how molecules move around in any substance. And then I happen to get close to this and I go, oh. And then I fall off it and I go back about my business, okay? But that means that overall, you've got less water movement occurring on this side of the tube. The direction of movement is entirely random, okay? But if there's more movement on the left side of the tube here, because there aren't as many solute particles to interact with, then more water molecules are randomly going to be crossing to the right. All right? So the net water movement ends up being in that direction, towards the area of higher solute concentration. Yes?
So is the reason why there's less water movement on the right side is because it's interacting more?
Yes, they're interacting more with these particles.
It's able to diffuse better.
Yes, because it's moving around more.
Okay, great.
It's moving around more, exactly. So that's why some particles are more osmotically active than others. A sodium ion attracts water pretty aggressively, but a molecule that does not have any regions of charge on it, all right, the water molecules aren't really going to be attracted, so it's not going to affect the kinetic energy of the molecules. So we usually don't talk about that in Bio 105. It's for another course, some course down the road that you might take. But I think it's relevant, okay? That we talk about osmosis in a very simple way. Water moves from areas of low to areas of high solute concentration. Water moves towards the hypertonic solution. But that is the pattern, not the principle or the mechanism. And so hopefully that brief aside that I just took gave you a little more insight into the mechanism. Now let's get back into the pattern and the terminology, which understanding the patterns and the terminology, that's the foundation of understanding any field. You can learn the mechanisms later. If none of that solute stuff sunk in, learn it later. Or don't. I get paid either way. All right? Solid. Tonicity, you talked about this in lab. Tonicity. It basically describes how much osmotically active solute is in a solution. A hypertonic solution has a bunch of osmotically active solute. Bless you. A hypotonic solution has a very low concentration of osmotically active solute. An isotonic solution has the same, but the same relative to what? Okay? Tonicity is always a relative discussion. Check it out. Let's flip back over here. In this example on the screen, this solution has a higher concentration of solute. It is hypertonic relative to this side of the tube. And this side of the tube has a lower concentration of solute. It is hypotonic relative to this side of the tube. After water has moved and the concentrations have equalized, now both sides are isotonic to one another. Question?
So tonicity is relevant, based off what it's being compared to?
Yes, 100%. So when we look right here at this cell, right, we've got this plant cell where there's a higher concentration of solute outside. The cell is floating in a hypertonic solution, and water flows out. But you could also describe the inside of the cell is hypotonic relative to the surrounding environment. It's all relative, but usually if we're talking about cells, we're talking about the solution the cell is floating in relative to the interior of the cell. Solid? Great question, points, very good. All right. Isotonic, there is movement going in, there is movement going out of the cell, but the net water movement is zero. Water's crossing the phospholipid bilayer because there's so much of it and it's a small molecule. It crosses the hydrophobic domain of the phospholipid bilayer, which remember is the primary barrier to movement across the cell membrane. Cool? Isotonic. Hypertonic. Higher concentration of solute draws water out of the cell. causes this animal cell to become shriveled, causes this plant cell to become plasmolized, which you should have seen in your previous lab. Yes, you looked at the onion cells. Here's what happens, right? The fluid leaves the cell, the plasma membrane gets drawn in from the cell wall. But remember our talk about cell membranes and how there's all these proteins that help to, say, anchor a cell to the extracellular matrix? Well, the same thing is true in a plant cell, and that cell membrane is going to be anchored to the cell wall, okay? So you got these anchor points, and this whole thing's shriveling in, but the cell doesn't entirely collapse. It's a special, state that we call plasmolized. Awesome. Next, hypotonic solution. Hypotonic solution, plant cells love it, okay? Because it lets them become turgid. You get internal pressure. That's what the plant cell wants as an overgeneralization. The plant cell wants to have positive internal pressure. It wants to be drawing more water in than is leaving. Now, to kind of get to your question there about gravity earlier, there are multiple components to water potentials. We're only talking about solute potentials here, all right? But pressure is something as well. So there's this whole concept of discussing water movements in terms of the energy that a solution has. Check this out, right? There's water in this bottle. I'm not gonna actually dump water all over the place, but you can imagine, all right? If I raise the water up, it now has more energy. It's potential energy. It has more energy of position. And I could pour it out and the water would fall out. If I flip the water down, it has less energy due to position, okay? Now, imagine I'm very powerful, and if I wanted to, I could crush this aluminum bottle, no problem, and blast the water all over the place. But I'm not gonna do that, So you squeeze the water bottle, you add enough pressure, and water movement goes out, all right? So check this out. Water's gonna flow into here, following the rules of osmosis, and eventually the pressure of the cell wall is going to counteract the water movement and lead to the net water movement becoming zero eventually, because more water is gonna get pushed out as water moves in. Anyways, it's all very fascinating. Osmosis and water potentials, usually discussions of pressure, position, all right, and then solute. Those are the main things we talk about. And then matrix potential in the soil. I'm gonna go 1, 2, 3. Cool? 1. Plants have cell walls.
So what about a cell that doesn't have a cell wall? Will it still become nice and explode?
Yes.
Okay.
And there are other things that have cell walls too, like fungi have cell walls, and and a lot of bacteria actually have cell walls too.
Need the cell wall to reach the requirement to become hypotonic.
That's the hypotonic, hypertonic, isotonic is all about the solution outside of the cell, right? Awesome. And number 2, yes, of course, yeah.
I was going to ask, could a solution ever be so hypotonic that even cells with cell walls explode?
I don't know of any examples of, no. Well, I don't want to just say no. It depends on the stability of the cell wall and blah, 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 blah. Like I have to give you all of the scientist talk where I say there's always an exception and all this, right? But if we're talking about a healthy, mature plant cell, then no. You can put it in pure water and it's not going to explode. The cell wall's very stable. If there's a weakness in the cell wall, or if it's a bacterium that has a weak cell wall already, yes, it could possibly lyse in pure water. But most things with cell walls, the cells won't lyse even in the presence of completely pure water, the most hypotonic solution possible, right? Cool? I love it, awesome. As a third hand right there, yes. This one? Yeah, totally. Yep, so this is in a hypertonic solution. Water goes out, plasmolized for the plant cell, shriveled for the animal cell. All right? Solid. You did the protein assay lab, right? Recently, that just happened, yeah? Where you exploded the cells or you shriveled the cells. Okay, cool. You're gonna do more osmosis with the potato. Do that yet? No, that's coming up, right? You did that this week? You did that one? It's hard to keep track of everything. I get so confused. Anyways, What did you do this week? You did some plates. You did some microscopy, right? You got to do gram staining. That's cool. That's pretty cool. Oh, you got to calculate the proportion of antibiotic-resistant bacteria. Yeah, nice. I like that activity. It's cool. Okay. How was this osmosis talk for everyone? Anybody have any questions about it? It's okay? Awesome. Osmosis is really, 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 really important. And water potentials are actually kind of fascinating if you're trying to predict where water is going to move. It's incredibly important if you're trying to predict if a biological or an ecological system is going to be stable. You need to know where the water is going to go. All right, you need to know if the plants can get at the water. So think about a root cell that is put, I'm just gonna keep going for a little bit. Think about a root cell that's growing into a soil that's got a lot of salt in it. What's gonna happen? The root cells are going to lose water because the surrounding solution is hypertonic. So the plant won't be able to access the water. That's why soil salinity is a problem, okay? Because of the osmotic pressure, all right, that gets generated. Just something to, think about. Cool. Man, I talk about a lot more osmosis stuff in my other classes. You take plant ecology, I'll talk your ear off about soils and water potentials. It'll be a lot of fun. Anyways, let's keep on moving forward. Here's a nice simple question. Mammalian blood contains the equivalent of 0.15 molar sodium chloride. So in every liter of sodium chloride, there are 0.15 moles. Remember, you'll never stop having to do this sort of math in this class where you have to convert from molarity to like grams of solute. So make sure you don't forget about it. It'll be on your lab midterm, it'll be on your lab final, everything, okay? But this is a pretty simple question where it's just saying, look, there's a lower concentration of sodium chloride in mammalian blood than in seawater. So if you put a red blood cell into seawater, well, that red blood cell is adapted to this lower concentration of solute, and you have put it into what type of solution? A hypertonic solution, exactly. Very good. So that red blood cell in a hypertonic solution, what's it going to do? Shrink, shrivel, A. All these are correct. Water will leave the cell, causing them to shrivel and collapse, right? Cool? Awesome. Now there's a couple of, sometimes the wrong answers are also informative. This one right here. NaCl will be exported from the red blood cells by facilitated diffusion. Why is that wrong? Yes? Okay, but remember, facilitated diffusion opens up a channel, and it would allow something else to diffuse. So that's the piece there. Good, yes?
Diffusion is a process that
Doesn't require energy.
So if it's facilitated diffusion, that would require energy?
Not quite. Facilitated diffusion just makes the hole, okay? So now there's a cell and there's a hole that would allow sodium chloride to move. Please. You can have 4 points. There you go. Points for you. Well done. In case you didn't hear it, diffusion goes from areas of high to areas of low concentration. So if there was a pore or a protein, a membrane protein, that allowed sodium chloride to diffuse, it would be going into the cell, not out of the cell. Awesome. Great. Okay, wrong. Wrong. Won't passively diffuse. Great. Cool. Awesome. Oh yeah, and then I go like, remember, passive transport, facilitated diffusion, here's what it looks like. Active transport, good. Okay, here's the sodium-potassium pump, which I need to honestly just kind of kick out of here because we don't talk about it enough and it ends up just being more confusing. But it's just meant to be an example of active transport, okay? So the sodium-potassium pump's incredibly important. Where is it? Here we go. Sodium potassium pumps, incredibly important in generating voltages. Animal cells need to generate voltages sometimes. If you want to transmit a signal really, really quickly throughout your body, you use an electrical current, okay? And you can generate voltages across membranes by having proton concentrations be different across membranes, all right? It kind of gets complicated because different molecules follow their own concentration gradients. It's not entirely based on charge. I'm going to try really hard not to just make this entirely too upsetting, too confusing. All right. So if you're pumping charged particles across a membrane, you're generating voltage. Okay. If you open up a membrane and just let, say, potassium flood into a cell, that can lead to a huge electrical pulse that then travels down, say, a nerve cell. We'll keep it simple. We're not going to talk about it too much. This right here is a proton pump. It's an electrogenic pump. And what it does is it transports sodium and potassium, but in opposite directions. So it sets up a high potassium concentration on one side and a high sodium concentration on the other side. Seriously, don't worry too much about it. You'll learn about it in your animal physiology course. If you take Zo 110, you'll learn about it there. That's way more important. We're going to be focused more on just simple proton pumps, okay? I would like you to focus on just simple proton pumps, where what they do is they consume some energy and they pump some protons. This is still active transport, okay? And it still generates voltage, okay? It still stores energy in a concentration gradient. So check this out. ATP gets consumed, protons get pumped. In this example, they're going from the cytoplasm to the extracellular fluid. You have a high concentration of protons here. You have a low concentration of protons here. This system wants to fall to equilibrium. These protons would love it if they could get across that phospholipid bilayer, but they can't because of the hydrophobic domain of the phospholipid bilayer, which is the primary barrier to movement. Right? I know, we love that. Okay, so this process is going to be way more important than you might think right now. Yes?
How do the hydrogen ions get inside the cell?
How did they initially get in? So water, right? Water is slightly reactive and it's constantly breaking apart and coming back together. So there's always going to be some concentration of hydrogen ions just in any aqueous solution, all right? Now, based on if you keep pumping those out, all right, you're going to reduce that concentration, okay? But there will always be some. Does that sort of make sense? Also, water can come from breaking down other molecules that have hydrogen and oxygen in them. Is that a satisfying enough answer? Awesome. Yeah.
Remember you mentioned before that it's really bad for ions to get diffused into the cell membrane, right?
Yeah.
Where do the hydrogen ions become dangerous over time?
Oh, yeah. If you get too much of these, it's acidic, you know? So if you pump enough of these protons into a cell or into an organelle, it becomes a bubble of acid, you know, and it can denature proteins. It can do all sorts of bad stuff. Yes. Satisfying? Awesome. Yes, please.
Does that lead to why cells need peroxisomes?
Not quite. Not quite. So the peroxisomes break down a slew of other molecules, but aren't necessarily involved in adjusting pHs.
Because they take out H2O2, right?
Yeah, hydrogen peroxide, which is a byproduct of breakdown of other molecules. So while the hydrogen peroxide is a dangerous byproduct that the peroxisomes will generate, they actually make it, and then there are other catalase enzymes that break that down. Those catalase enzymes are pretty widespread because we're actually making hydrogen peroxide a lot inside of our bodies. It's not quite connected to this concept. Solid? I love it. great. But being able to make chambers acidic is important. Think about the lysosomes, okay? Where there's specific, man, did I talk about that yet? It's really cool. Real quick, just bear with me, because it's interesting, all right? I'm just gonna talk to you about something interesting. A lysosome, right? What's a lysosome got inside it? It's got enzymes. What do we know about enzymes? Well, that they are proteins, so they can become denatured, all right? Enzymes have specific pHs where they operate really well. So what you'll do is you'll have a lysosome, and then there'll be some proton pumps that regulate the pH inside of the lysosome so that the enzymes can be active. But if the enzymes accidentally get released, because remember, these are enzymes that just break things down. That's what the lysosome's designed to do. If the enzymes get released and they go into the neutral pH of your cell, they get denatured. They don't work anymore, all right? So that's a way that your body protects you from if accidentally a lysosome breaks open, by making the lysosomal enzymes only work at certain pHs, and you can modify pHs of chambers by pumping protons, and the protons can't freely diffuse across the phospholipid bilayer. It's all connected, all right? Cool. So anyways, these proton pumps are great. We need them for carrying out photosynthesis. We need them for carrying out cellular respiration. They're incredibly important. All you need to know about them for now is that they consume energy and they pump protons to set up a proton concentration gradient. And let me just add to that, that concentration gradient can be harvested for energy. Okay. Moving on, this is more membrane stuff. We already talked about endocytosis. We talked about phagocytosis, but endocytosis and exocytosis, which isn't on here. Good. That's good. My bad. All right. Endocytosis is when you fold in your membrane, all right? You make a vacuole to bring something in, all right? Cool? Phagocytosis is cellular eating, where you bring in a food thing. Penocytosis is cellular drinking, where you bring in some extracellular fluid, all right? Receptor-mediated endosymbiosis, or endocytosis, receptor-mediated endocytosis, it relies on these receptor proteins that bind something specific, and then the cell goes, oh, I've bound the thing that I wanted to these receptors. Now they're already captured on the receptors. Let me just fold them in to a little vacuole, all right, or a vesicle, okay? And now they're trapped. Now I have what I want, and everything's already bound to the receptors. You see how that sort of works? Now perfect. It's hard to, I wish my hands would bend that way, but they just don't, okay? So check it out. Phagocytosis, penocytosis, receptor-mediated endocytosis. There's one question that I use, I don't know, I think I use it every other semester, or it might be on a reading quiz or something, about where the outside of the cell ends up following endocytosis. And it's the inside of the vesicle or the vacuole, okay? Check it out right here. Here are the receptors. They receive the signal, they bind their ligand, all right? And then the membrane folds in. So what was the outside of the cell is now the inside of the vesicle. Does that make sense? And what was the inside of the cell is now the outside of the vesicle. If you continue in cell biology, this is important, you know? But it's also a question about reasoning. You have to understand what endocytosis is in order for this. If you understand what endocytosis is, that question makes sense, you know? Like it was the outside, and then it folded in. It was the outside, and then it folded in, okay? So now it's the inside. The tops of my hands are the outside of the cell. They fold in to capture something, and now they're the inside of the vesicle. Fair? All right, I love it. So that's another way that things can cross membranes. That's how it ties to anything else. It's like, how does all of this tie together, right? It's all about what can cross a membrane, freely, how do proteins help things cross membranes, And hey, if something's really big or you want to move a bunch of fluid all at once and you don't care about specific stuff, then just make a vesicle or make a food vacuole and pull the stuff in. All right? Pull a bunch of extracellular fluid in. Does that sort of tie it all together? This is, I think, the first time I've got one of these flow charts on the screen for you. But you'll see in future lectures, I use these flow charts to try to tie a whole lecture together. Okay? And we'll just fill it out together. You excited? I know you are. Me too. Me too. Yeah. Yeah, here we go. Molecules can cross cell membranes by two categories of process. I mean, we're not including endocytosis here, but we could also have another category of endocytosis. But don't worry. We won't worry about that. Molecules cross cell membranes. How? By passive transport and by... I'm saying it. Oops. Okay. Passive transport moves down blank. Active transport moves against blank. What? Concentration gradients. Awesome. Okay. Passive transport, it could be diffusion, or it could be... That's good, but osmosis is diffusion, really. What other way do things diffuse that wouldn't freely cross the phospholipid bilayer? Facilitated diffusion. Whoever said that gets points. Points. Who said that? I'm very proud of you. Very good. Good answer. Correct. All right. Facilitated diffusion. All right. So facilitated diffusion will be of polar molecules and ions, things that wouldn't normally cross the phospholipid bilayer. Regular old diffusion like this, what does that work with? Hydrophobic, exactly. Boom, points. All right, hydrophobic molecules, small uncharged polar molecules, hydrophobic molecules, small hydrophobic molecules are the fastest, right? Bless you, they're gonna go the quickest across. Cool. What do we got here? Cross cell membranes by active transport. Active transport requires ATP. Now both facilitated diffusion and active transport use what? Membrane proteins, exactly. I love it. Great job, everybody. We filled out the dang flowchart. And we got through the week 5 material on Friday of week 6. That's pretty good. That's pretty good. Let's just rock it through the week 6 PowerPoint. What, I think it's like 60 slides, 52 slides. We'll just do it right now. Ready? And go. All right. Did you get it? I'm so glad you enjoyed it. Yeah, I'm proud of you all. Huh. All right, let's just spend, well, we got 13 minutes left with each other, 12 minutes left. Let's start talking about energetics. It's kind of fun stuff. It's a nice way to think about the world. And, remember, I talk about how different models work for different people, different fields, at different scales. The physicist, the chemist, the biologist, they all look at the same thing in different ways. And we're going to put on our chemist and our physicist goggles for this discussion of energetics. We're going to really be approaching this more as physicists than as chemists or biologists. But that's okay, all right? Let's talk about the terminology that we use. Metabolism, catabolism, anabolism, all right? What does any of it mean? Well, metabolism is just the name we give for all the chemical reactions going on, all right? It can be all the chemical reactions going on inside of a cell. It can be all the chemical reactions going on inside of an organism. So when people say, oh, I have a high metabolism or I have a low metabolism, what they mean is that they have rapid rates of all the chemical reactions going on in their body, or they have low rates of all the chemical reactions going on in their body. Fair enough? Now, metabolic reactions are broken into categories based upon whether they build things, whether they break things down, whether they require energy, or whether they release energy. We'll talk about them. I'll give you good mnemonic devices, and you'll be comfortable by the end of these, probably to the end of this lecture, talking about anabolic and catabolic reactions. So, Anabolic reactions, what do? They take little things and they build bigger things. All right? Catabolic reactions, what do? They take bigger things, they break them down into littler things. Okay? Now, I got my water bottles. Here we go. These are all, when these come out, the real learning's about to begin. So check it out. These are both now, whoa, when I tapped it, it moved my phone. The angle's not right. I'm gonna fix that. All right, hello. Okay, solid. So these are now monomers, all right? These are now subunits of something that's gonna be built. If I wanna make them into a polymer, I have to invest energy. So here I go, I invest energy, and now I've created more order, all right? I have fought against entropy. I have created a larger molecule, and I've consumed energy in the process. Now, that structure now holds energy. And look, just looking at it, it feels less stable, right? That feels less stable than the two monomers sitting next to each other. So there's energy held in this, really in the position, all right? But if we're talking about the chemical analog, there's energy here in the bond, all right, between these. And if you can break that bond, you can release the energy, right? If I knock this down, it goes clang, bang, and it releases a bunch of energy, right? So I invest energy here. That's an anabolic reaction. The energy gets stored, at least some of it, in the structure of the larger molecule. And then when I come over and I break that bond, the energy gets released, okay? That's a catabolic reaction. A large molecule is broken down into smaller molecules, and the energy gets released. Now, every time you convert you go between these, you lose energy, right? You don't get all the energy I put into here to make the molecule doesn't all come out when I break the molecule, all right? But that's catabolic and anabolic. Question, please.
You said for the
Anabolic.
sorry.
No.
That the larger molecule has more order but is less stable.
Yeah.
Right? Well, I'm like, my brain's like order and stability should go together. So how come it has more order but less stability?
Totally.
This goes into entropy.
This exactly goes into entropy.
Okay, then
Yeah.
we'll learn about it in a second.
Yep, exactly. We're going to talk about it in just a second. Solid. I promise. And it's because we have a common vernacular for discussing order, and then we have the physicist way of discussing order, and so it gets tricky. We should use an entirely different language for sciences, and then we wouldn't have to worry about any of this, confusion. We just have to learn a different language for each field. Why not? Okay. Here we are. Okay, cool. So anabolic, catabolic reactions. All right. Questions about these two broad categories and what they describe? I'm stoked to hear it. Let's look at this classic example of a catabolic reaction. Okay? You got the gasoline molecule. It's got a lot of structure. It's got a lot of order to it. You break it down in a catabolic reaction, okay, by burning it, letting it explode. You get these smaller molecules. They are less ordered, all right, and you release a bunch of energy. Cool? So, catabolic reaction, super standard. Some of that energy gets converted into energy of movement. Some of that energy gets lost out of the system as heat energy. That's what is it? That's the second law of thermodynamics or the first one? We'll recover them in a second. Look, in the cell, it's like the same thing, except the cell doesn't have wheels. All right? Check it out. You break down glucose, you produce carbon dioxide and water. Cellular respiration occurs, and instead of converting the energy that was in glucose into kinetic energy of movement, you are instead generating ATP, a temporary energy currency. which then is going to be consumed later to carry out cellular work, some of which will be kinetic, like moving a dang motor protein along a microtubule, like we talked about. Okay? Solid. Catabolic reactions, both of them. Energy. Let's talk about energy. We have to talk about potential energy, kinetic energy. It's thrilling stuff, okay? Look at these penguins. They have potential energy by standing up here, and they can harvest that potential energy by sliding down and jumping. They convert potential energy into kinetic energy. Blah, blah, blah. You've seen this example before. I take the ball, I put it on the ramp, I roll it down the ramp, look at it convert potential to kinetic energy. Fine. But potential energy, also, it's all about the position of things, right? So I invest energy into this water bottle, and I give it more potential energy by making it higher up, right? The position of electrons, the position of atoms in a molecule, these are also examples of potential energy, okay? So energy stored in the location of or structure of matter. So that's the macro scale of I stand up here and I gain potential energy, okay? And also the micro scale of a plant carries out photosynthesis and makes a complicated organic molecule. That complicated organic molecule has potential energy that is stored in the location or structure of matter. So it is still potential energy, but if it's in at a molecular scale, we call it chemical energy. How's that feel? There's going to be a time when I'm talking to you about electrons going from a high to a low energy state, and it's really analogous. It's really analogous to this, right? Low energy state, put it at a high energy state, let it fall to a low energy state, harvest the energy. Raise it to a high energy state, invest some energy. Let it fall to a low energy state, harvest some energy. All right, this is now an electron. Yes?
So it's kind of like pedaling on a bike.
Yeah, kind of, yeah. You put the energy, it's like taking a bike up a hill.
Okay.
You know, invest, 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 harvest. Yeah. So yeah, it's like pedaling a bike. Okay. Energy can neither be created nor destroyed. It just changes forms. We've heard about this before. All right, the laws of thermodynamics. Two big ones. Can't create energy, can't destroy energy. There's like a net pool of energy, and energy just changes whatever form it's in. And every time energy changes form, you lose a little bit of energy as heat. And that just sort of radiates out into the system. And what is its fate? Don't worry about it. All right? So, like I said before, every time I change energy between different forms, you lose a little bit. So you can't harvest everything. Good. So we've got this overall, big picture bioenergetics thing going on right here. Solar energy blasts the planet. Photoautotrophs, they convert that solar energy into organic compounds, which have potential energy. They also produce oxygen, because these organic compounds are carbon-based molecules. That's why they're organic. And they get the carbon from carbon dioxide to build all those carbon skeletons. Okay? So when they break carbon off and use it to build these organic compounds, they release oxygen. More energy here, all right? And then the chemotrophs, or the heterotrophs, all right, consume that high energy organic molecule, break it down in a catabolic reaction to carbon dioxide and water. They release all that energy to drive their own production of biomass and all their day-to-day activities. In both of these, you're converting energy between forms, so sure enough, you're losing some as heat due to inefficiencies in the system. All right, bioenergetics, yes?
I just realized, could you technically just say all foods are organic because they're made of organic molecules?
100%, yeah, absolutely. Absolutely, you can say that. Solid. Anyways, that's the end of our time today. I really appreciate you making it to Friday of week 6. This is like when everything starts to feel hopeless, and I totally get it. But before you know it, it'll be week 10, okay? And in week 10, life gets good again, all right? So just make it through a month with me. We'll have a good time.